This PowerPoint will focus on one of the central metabolic pathways, glycolysis. Remember that the central metabolic pathways are amphibolic, having catabolic and anabolic purposes. We're going to focus on the catabolic functions of glycolysis. Glycolysis functions will be to provide energy in the form of reducing power. It will rob hydrogens from glucose, giving those hydrogens to electron carriers, mainly NADH. Glycolysis will also produce precursor metabolites, amino acids and monosaccharides. Glycolysis will also produce immediate use energy in the form of ATP by a process called substrate level phosphorylation. So these will be the three catabolic end results of glycolysis. So the purpose of glycolysis is to oxidize, remove electrons from glucose, and in the process, two molecules of a 3-carbon compound called pyruvate will be produced. Glycolysis takes place in the cytoplasm or cytosol of eukaryotic and prokaryotic cells. Glycolysis is a universal pathway present in virtually every single living cell. Glycolysis will involve splitting a 6-carbon glucose into two 3-carbon compounds, pyruvic acid. Glycolysis has two steps. The energy investment stage, which requires energy in the form of ATP, and an energy generating stage, which creates energy in the form of ATP. The first steps in glycolysis are the energy investment steps. During these steps, glucose, a 6-carbon monosaccharide, is phosphorylated using an ATP molecule. So ATP will be hydrolyzed into ADP. ATP loses a phosphate. The phosphate attaches to glucose, and the product of the reaction will be glucose 6-phosphate with the phosphate attached to sugar carbon number 6 of the sugar. The second step in the energy investment stage of glycolysis will involve the rearrangement of the glucose molecule into a fructose molecule. Fructose is also a monosaccharide. Notice that the phosphate group is still attached, making fructose 6-phosphate. The third stage in the energy investment stage of glycolysis will require another phosphorylation of the fructose molecule. So ATP will be hydrolyzed into ADP. The lost phosphate will be attached to carbon number 1 of the fructose, creating fructose 1,6-biphosphate. What we now have is a molecule that can be easily split because it has two unstable phosphate groups that will allow the molecule to split into two 3-carbon compounds. That will be the last stage in the energy investment stage of glycolysis. Fructose 1,6-biphosphate will be split into two 3-carbon compounds. Notice that the two 3-carbon compounds are not identical. One is called G3P or glyceraldehyde 3-phosphate. The other one is DHA or dihydroxyacetone. Notice that each, however, will have a phosphate group attached to it. So the first step in the energy investment stage of glycolysis involves taking glucose and phosphorylating it using an ATP molecule. The phosphate group will be attached to carbon number 6 of the glucose. So the first step involves converting glucose into glucose 6-phosphate. The enzyme that catalyzes the reaction is hexokinase. During this first step in the energy investment step of glycolysis, ATP is hydrolyzed to ADP. The second step involves changing glucose 6-phosphate into fructose 6-phosphate. The conversion takes place via the enzyme phosphoglucose isomerase. The third step involves again another phosphorylation. ATP will be spent, hydrolyzed into ADP, and the phosphate will be attached to the first carbon of fructose, changing fructose 6-phosphate into fructose 1,6-biphosphate. Fructose will now have two phosphate groups, one attached to carbon number 1, the second one attached to carbon number 2. This is the second ATP hydrolyzed during the energy investing steps of glycolysis. Now the end result is a molecule that can be easily split into two 3-carbon compounds, the phosphate groups providing the unstable groups that allowed for the splitting of fructose to take place. So the last step in the energy investing step of glycolysis is the splitting of fructose 1,6-biphosphate into two 3-carbon compounds, G3P and DHAP. G3P has 3 carbons, 1, 2, 3, with a phosphate attached to carbon number 3. DHAP also has 3 carbons, 1, 2, 3, with a phosphate attached to carbon number 1. DHAP will be changed to G3P. Thus, the end result of the energy investing step in glycolysis will be 2 G3P molecules. The energy investing stage in glycolysis ends with 2 G3P molecules, which were the product of splitting one 6-carbon molecule of fructose. Now starts the energy generating stage of glycolysis. So far, 2 ATPs have been spent. No ATPs have been produced. During the energy generating stage of glycolysis, each of the G3P glyceraldehyde 3-phosphate molecules will undergo an oxidation step, the removal of hydrogens, the dephosphorylation steps that will create ATPs, 2 dephosphorylation steps. The end result will be 3-carbon compounds called pyruvate. Each of the G3Ps will undergo oxidation, 2 dephosphorylations that will end in the formation of a 3-carbon compound called pyruvate. The first oxidation step during the energy generating step of glycolysis involves the removal of hydrogen atoms from G3P and the reduction of NAD+ into NADH. Now we have one electron carrier molecule being produced. Later in the steps, 2 ADPs will be phosphorylated into 2 ATPs. So 2 ATPs will be generated per G3P molecule. The end result will be a very stable molecule called pyruvic acid. So each G3P molecule generated during the energy investment stage will undergo an oxidation step, creating 2 NADH molecules total and 4 phosphorylation steps, creating 4 ATP molecules total. The dephosphorylation steps are the step in which 1,3-biphosphoglycerate is dephosphorylated, the phosphate is given to ADP, and ATP is produced in the process. The enzyme in the reaction is phosphoglycerate kinase. The substrate is the source of the phosphate that is given to ADP, which is then converted to high-energy ATP. The second dephosphorylation step occurs at the end of the energy-generating stage. When phosphoenopyruvate is dephosphorylated, the phosphate group is given to ADP, which then turns into high-energy ATP. Pyruvate is the stable product of the reaction. Pyruvate kinase is the enzyme that catalyzes this step. In both these steps, ATP is produced by a process called substrate-level phosphorylation. It is called substrate-level phosphorylation because the phosphate given to ADP comes from the substrate of the enzyme involved in the reaction. Again, the phosphate given to ADP to make ATP is coming from the substrate for the enzyme in the reaction. So glycolysis produces ATP by a process called substrate-level phosphorylation. One of the most important facts that you need to glean from glycolysis are the end products of glycolysis. The end result of glycolysis is the formation of 2 NADH molecules, 4 ATP molecules, and 2 pyruvate molecules. In summary, glycolysis will take a molecule of glucose and split it into 2 G3P molecules, which will later be changed into 2 pyruvate molecules. So one 6-carbon compound will be turned into 2 3-carbon compounds. In the process, 4 ATPs will be generated per glucose molecule. However, remember that 2 ATPs were spent during the energy investment stage of the process, giving us a net ATP gain of 2 per glucose molecule. Also, 2 NADH oxidation steps will be provided per glucose molecule, and 2 pyruvic acid molecules will be the product of glycolysis per glucose molecule. The central pathways like glycolysis are amphibolic pathways. The catabolic steps in this pathway provide energy in the form of reducing power. In the case of glycolysis, 2 NADHs are produced. Also, precursor metabolites will be produced. Pyruvic acids are precursor metabolites which can be used to create amino acids. Immediate use energy is formed in the creation of a net gain of 2 ATP molecules. You should now be able to fill in the pathway chart for chapter 6, in which the location of the pathway, in this case glycolysis, in eukaryotes and prokaryotes is asked. Also, you should know the reactants, the starting reactants in glycolysis, the final product of glycolysis, the number of oxidation steps per glucose molecule, or the number of NADHs produced, the net number of ATP synthesized per glucose molecule. The number of decarboxylation steps are puzzling because Decarboxylation steps are steps in which carbons are removed. Are there any CO2 produced during glycolysis? We start out with one 6-carbon compound. We end with two 3-carbon compounds. Have there any carbons been lost during glycolysis? Hopefully the answer is obvious.